All right, Mr. Lally, your next witness, please.
Yes, Your Honor. The Commonwealth call Ms. Tess Chart to the stand.
Okay.
Just step right up here and just watch your step.
Why in the courtroom, please?
Let me stand and face the clerk.
You raise your right hand. Do you solemnly swear the honesty of the court and jury in the case down here and the truth, the whole truth, and nothing but the truth, so help you God?
I do.
Thank you. You may take the stand.
Go right ahead, Mr. Lally.
Thank you, Your Honor. Good afternoon, ma'am.
Good afternoon.
Could you please state your name and spell your last name for the jury?
Sure. My name is Tess Chart, C-H-A-R-T.
And Ms. Chart, what do you do for work?
I'm a forensic DNA analyst at Bodhi Technology.
And where is Bodhi Technology located?
It's in Lorton, Virginia.
And I'll ask you some questions, just a little bit about your educational background, starting with your undergraduate work. Where did you go to school, and what, if any, degree did you receive in relation to your undergraduate?
Sure. I have my bachelor's in science in molecular biology from Millersville University in Pennsylvania. And then I have my master's of science in forensic science from Towson University in Maryland.
All right, so, Ms. Chart, I'm going to ask you to keep your voice up really loud, okay? Thank you.
Ms. Chart, if it's at all helpful, that microphone is adjustable. You can move it anywhere. Now, with reference to your master's in forensic science, when about was that you received?
So I got my degree at the end of 2019.
And following grad school, where did you go from
I was hired directly by Bodhi Technology.
there? And when was that?
That was in July of 2019.
Now, when you started in July of 2019 with Bodhi Technology, what was your position at that time, and sort of what were your duties and responsibilities when you initially began work?
So I started as a technologist. So that's someone that will do all of the lab portion of the work in a laboratory. And I helped out with our humanitarian team, so helping identify human remains. From there, I also helped with our criminal cases as well.
And when you started your work there, what, if any, sort of training did you undergo in relation to your work at Bodhi?
So when you first start at Bodhi Technology, you actually go through a series of lectures and you have a series of readings to go through all of the science that you're going to learn and the background of all of the technologies you're going to use. From there, you will observe a qualified analyst or a qualified person in the lab, go through everything that you're going to be trained in. And from there, you'll get a series of sets. So there'll be mock samples that you will take through the lab and do all of the testing on that you're going to be trained in. And it starts out with having that qualified analyst there with you, and you're able to ask questions and troubleshoot as you go. From there, you'll get a second set that'll be no analyst with you, but you're still allowed to ask questions at that point. And then your final set is something that we call a competency set. So you take it through the lab and treat it just like you would any other case. And you're not allowed to ask questions, and then you're graded at the end. If you pass that test, you can go through a written exam and then a verbal exam. And then you take your results from your testing and you defend it in a mock court sample. If you pass all of those qualifications, you can become an analyst or become qualified in whatever lab technique you're being trained in.
And as far as that sort of training process went, how long was it from the time that you joined Bodhi Technology to the time that you started doing the work that you do now?
It's been a rolling process of all the different trainings that I've had to go through. I'd say it took me about 9 months from when I started to be qualified in everything that I do for mitochondrial DNA testing. And then for analysis training, I think it was another 6 months or so.
Now, as far as the lab at Bodhi Technology, is that an accredited lab?
Yes, it is.
And do you know from whom the lab receives its accreditation?
Yes, we receive a international accreditation from an association called ANAB, which is just a national standards organization. We're also accredited by the FBI at their quality standards as well.
If you could just keep your voice up, a bit, please.
Would you like me to repeat that?
No.
Okay.
Ms. Chard, as far as your lab being accredited, can you explain to the jury sort of what that means and sort of what the process is for lab to be accredited?
Sure. So all of our testing, all of the things that we report are basically guidelined by an outside body. So everything we do is established and gone through before or as we go through the testing procedures. So we're able to make sure that everything that we do is standardized for the country, all the testing that we perform.
And the lab that you work at Bodhi Technology, is that accreditation up to grade at this point?
It is.
Now, with reference to your own work within the lab, is there sort of standards of procedure or protocols that you follow in conformity with that accreditation?
We're under the same standards as all other DNA testing at Bodhi for mitochondrial DNA testing.
Now, with regard to your specific work, are you familiar with what's called a proficiency test?
Yes, I am.
Can you explain to the jury what that is and what your experience has been with proficiency testing as far as how often and what it consists of?
So a proficiency test is how we stay qualified in what we do in the laboratory. So we get an outside test that we treat just like a regular criminal case. We take it all the way through the laboratory and have it reviewed, report our findings, and then it's sent out and we're graded on that performance.
And with reference to yourself, what, if any, issues have you had with regard to proficiency testing over the course of your years of work with Bodhi Technology?
I've had none.
Now, as far as, am I correct in that Bodhi Technology, as far as the lab is concerned, essentially deals with being contracted for work from external vendors?
Yes.
By that, I mean as far as you get your work or samples that are submitted from outside of your lab, correct?
We do.
And what are some of the types of organizations that, as far as the casework that you deal with, like where does it typically come from? What type of agency?
It can come from anywhere across the United States, and we also have international clients as well.
Now, if I could turn your attention to a specific case that you worked on. When items come into Bodhi Technology, as far as the lab, they go through sort of a check-in process and a chain of custody process. Is that correct?
Yes, that's correct.
And with that, is there sort of a number or something that's assigned to a specific sample that follows it throughout the time that it's at your lab?
Yes, we'll call that the evidence item number, or E- whatever number it's assigned.
Is there also a case number that's assigned as far as different cases or different case submissions?
Yes, there is.
And turning your attention to a specific case that was submitted in January or partially submitted in January and then partially submitted in March of 2024. That came from the Norfolk District Attorney's Office. Are you familiar with that?
Yes, I am.
And the Bodhi case number on that was CCA 2416-0023. Is that correct?
That is correct.
Now, with reference to your work on this specific case number, what were the items that you received and sort of what were the items that you received that you eventually did some testing or analysis on?
So I continued testing for one of the hair samples.
And specifically, again, that's given a specific name as far as in the hair sample in this case was EO1, is that correct?
That is correct.
And then as far as for comparison purposes, what if anything did you receive in regard to comparison if you were able to generate a profile from EO1?
We received a reference profile from the victim, John O'Keefe.
Now, your specialty within the lab is in mitochondrial DNA testing, is that correct?
Yes.
Could you explain to the jury as far as mitochondrial DNA? Let me ask you this first. Are you also familiar with the term known as autosomal DNA?
I am, yes.
If you could, Ms. Chard, explain to the jury first what autosomal DNA is, and then what mitochondrial DNA is, and then I'll have another question.
Of course. So what many people are familiar with is what we call autosomal DNA. So that's the DNA that's actually stored in the nucleus of your cell. You get half from your mother and half from your father, and it's what makes you unique. And it's what people call the blueprint of a person. But what might be a new concept is that there's actually a second form of DNA that is stored within the mitochondria of a cell. So if you can remember maybe back to any science classes that you had, the mitochondria is the powerhouse of that cell. And because of that, you can have upwards of hundreds to thousands of mitochondria within just one single cell. And within each of those mitochondria, you may have thousands of copies of that mitochondrial DNA. So in cases just like this one, where there may not be enough nuclear or autosomal DNA to find, you can go back and try to test for that mitochondrial DNA.
And so if you could explain to the jury some, I think you've alluded to it already, but sort of the difference between autosomal DNA versus mitochondrial.
So mitochondrial DNA, in contrast, is inherited directly from your mother. So you will have the same mitochondrial DNA profile as your mother, and then you will also have the same mitochondrial DNA profile as any of your siblings. And it has that maternal inheritance that we call it, that makes it actually a great source for helping identify people, but it can also be used for criminal cases as well.
First, with respect to the autosomal DNA, can you explain to the jury sort of the process as far as just in general terms? What is the process as far as the testing goes or generation of the profile?
Sure. So we share similar processing methods. So we will start out by taking a small portion of whatever is sent to the lab, and we'll call that sampling. We'll take notes on that and how much that we take forward through testing, and that will go into a small tube. From there, we will extract the DNA or take apart all of the cells by adding chemicals. And what we're left with is what we call a DNA extract. So that's actually where I came in this case. From there, the difference is we will go through what is still called amplification using the PCR method. That will make a bunch of copies of that DNA to be used later on. But for mitochondrial DNA, what we're targeting is specifically just the mitochondrial DNA in that extract. From there, we actually go to something that is completely different from autosomal testing, which is DNA sequencing. So DNA sequencing is really just a way that we are able to take those small strands of DNA that we've amplified, and we're able to read them almost like letters in a sentence.
And the PCR is in reference to the autosomal DNA, is that correct?
It's for both methods.
What does PCR stand for?
It's a polymerase chain reaction.
And when you say as far as the sort of quantification, I'm sorry, is it quantification or quantitation?
Quantification works.
As far as the quantification step is concerned, how is that sort of physically done? What kind of instrumentality are you using to accomplish it?
So for mitochondrial DNA, we will actually run the DNA after amplification on a gel, so a long, really what it sounds like, slab of gel, where we're able to visualize if we obtained any mitochondrial DNA as a band that shows up under a camera.
So with reference to this sample, this EO1, the hair sample, you received it at what stage again, or what step?
I received it as a DNA extract, so it was a amount of liquid in a tube at that point.
And as far as, in general terms, again, if you could explain to the jury, so what are the steps that go into the processing or analysis prior to that step where you received it on the quantifications?
So it went through sampling, it was received, it was inventoried by the person that opened up the evidence, and a small portion or the hair was put into a tube, and then it went through that DNA extraction. So the hair was dissolved and all of the DNA inside of that hair was released and isolated to just be then that DNA extract.
So from that point where you pick it up as sort of the extract, what is it that you're doing with that as far as your analysis goes from there, and what type of instrumentality are you using to perform that analysis?
So from there, I am targeting and just trying to find if there is any mitochondrial DNA in that sample. So we'll use the PCR method to amplify to see if there is any mitochondrial DNA we can target. And then I will take it through and visualize it on that gel to make sure to see if we got anything. That is our quantification step. And then after that, I will take it through our sequencing step. And we do load that on an instrument called the 3130 genetic analyzer.
And the 3130 genetic analyzer, how does that work?
So it actually runs and separates each of the fragments of DNA that we have amplified and visualizes it through a program. So we're able to read each letter of your DNA sequence.
I could take you back just a step. Are you familiar with a term within your field called a D-loop?
Yes.
Can you explain to the jury what a D-loop is and how that factors into your analysis or testing for mitochondria?
So the D-loop is actually the region of the mitochondrial genome that we target. It is targeted because it does not contain any useful areas for the actual function of a mitochondria. So it can change over time, and those changes are what actually are recorded as your mitochondrial DNA profile.
Now, as far as the D-loop, what, if any, relationship does that have with respect to certain variables within the human population?
Would you be able to word that?
Very poorly put. Let me rephrase. The D-loop, as far as the region is concerned, what, if anything, does that have to do with certain variabilities within the human population?
Oh, yes. So because there is this variability in that region, it can change over time. And from that, there are different mitochondrial DNA profiles that are developed across the world.
Now, in addition to the extract from the hair sample EO1, what, if any, other items did you receive in regard to comparison?
So we also received that reference sample from the victim, and that was taken forward for mitochondrial DNA testing as well.
And just for clarity purposes, when you say reference sample from the victim, you're referring to John O'Keefe, is that correct?
Yes.
And the reference sample that you received in regard to John O'Keefe, was that already an extract as well, or what, if anything, did you have to do with that in order to obtain a mitochondrial DNA profile?
So that sample was received, if I can refer to my notes. I did not. So I was not the person to actually do the sampling for this sample. It was received as a small cutting of a swab.
And so, as far as the small cutting from the swab, what is sort of, as far as creating a profile from that, creating a profile from the extract from the hair sample as well, was there any difference in that, sort of how those two mitochondrial DNA profiles were created?
No, they were tested the same way.
And well, let me ask this, as far as with regard to the hair sample EO1, were you able to obtain a mitochondrial DNA profile for the hair?
We were.
And with respect to Mr. O'Keefe's sample, were you also able to obtain a mitochondrial DNA profile from Mr. O'Keefe's sample?
Yes, we were.
Now, as far as for comparison purposes, what, if anything, did you look at, and what, if any, comparison or opinions or conclusions did you draw from that comparative analysis?
Sure, I can state directly from my report if that's all right.
Okay.
So the mitochondrial DNA profile that was obtained from the sample, the hair sample, was consistent with the mitochondrial DNA profile obtained from John O'Keefe. So therefore, John O'Keefe cannot be excluded as a possible contributor of that hair.
Now, Ms. Chart, are you also familiar with something called an MPOP, and for the record, that's EMPOP?
I am.
And can you explain to the jury what that is and how that's used as far as any sort of statistical analysis in reference to comparative comparison?
So it's a international populations database that houses different mitochondrial DNA profiles.
And how is it sort of used within your field, and how did you use it in this specific instance in relation to the consistency opinion between the hair sample and Mr. O'Keefe's sample?
So we were able to upload the profile from the hair and see if it was found at all within the population database. We test populations that are popular within the United States to narrow it down. And within the African American, US Caucasian, and US Hispanic databases that they have, it was not seen.
And what, if any, conclusions or opinions are you then able to formulate with reference to the consistency of the hair sample versus Mr. O'Keefe's sample with relation to that MPOP data?
Sure, so by not seeing it in the database, this means that we're able to exclude at least 99.895% of the population from being a source of that evidence.
And is there a certain level or percentage of confidence statistically that comes along with that exclusion of 99.895% of the US population?
Yes, we use what is called the 95% confidence interval. It's actually a rule that is used for making statistical statements about populations. It's because there are always outliers that we don't include. We take the 95% majority of a population when we run those statistics.
And so, again, as far as your opinion or conclusions based on the comparison, is that the mitochondrial DNA profile from the hair sample was consistent with the mitochondrial DNA profile from Mr. O'Keefe's sample, correct?
Correct.
And that's with a 95% confidence, is that correct?
That is correct.
And that's the exclusion of at least 99.895% of the population of the United States, correct?
Correct.
Thank you, ma'am. I have nothing further.
We have no questions, Your Honor.
All right, Ms. Chad, you are all set. Thank you very much.
Thank you, Your Honor.
Your next witness, Mr. Lally?
Yes, Your Honor. The Commonwealth will call Mr. Andre Porto to the stand.
Look right this way. Just watch your step. Okay. stand and face the clerk.
Raise your right hand. Do solemnly swear the evidence you give the court and jury in the case down here and the truth, the whole truth, and nothing but the truth, so help you God. You can take the stand.
Whenever you're ready, Mr. Lally.
Thank you. Good afternoon, sir.
Good afternoon.
Um, First, let me just say that, just for clarification, that microphone in front of you is adjustable. You can move it up and
Okay,
down
cool.
any which way. Good afternoon, sir. Could you please state your name and spell your last name?
Yeah, my name is Andre Porto, last name spelled Porto.
And what do you do for work, sir?
I'm a forensic scientist too at the Massachusetts State Police Crime Lab in the DNA unit.
And sir, if I could ask you just a few questions about your educational background, starting with your undergraduate work. Where did you go to school, and what, if any, degrees did you receive from that?
I received a Bachelor of Science in Microbiology at the University of Massachusetts in Amherst. And then,
And where did you go from there?
I went to the Boston University School of Medicine and I got a Master of Science in Biomedical Forensic Sciences.
And about what year was that you received that master's?
I graduated 2019.
And then following receipt of your master's, where did you go from
I went to the DNA unit in the Massachusetts State Police Crime Lab.
there? And when you joined the Massachusetts State Police Crime Lab initially, what was your title and sort of what was your duties and responsibilities with relation to that title?
So I joined as a Forensic Scientist 1, and that's a training position where I underwent a training program that consisted of a bibliography with multiple journal articles and textbooks, as well as conducting the lab processing on training samples.
And that sort of initial work that you did as referred to as an FS1, is that correct?
Yes.
And that initial work that you did as an FS1, how long did you do that before you became a forensic scientist 2?
Approximately, the training itself was approximately 6 to 8 months. And then within a year, I was promoted to an FS2.
And when you were promoted to work as an FS2, what, if any, specific unit were you assigned to within the lab?
The DNA unit.
And have you essentially been with the DNA unit since that assignment initially occurred?
Yes.
Now, just a few questions about the lab at the State Police Crime Laboratory. Is that lab accredited by anybody?
Yes, we are accredited by ANAB, and that stands for ANSI, National Accreditation Board, and the ANSI stands for American National Standards Institute.
And with reference to that accreditation, can you describe to the jury just a little bit about generally in terms of what goes into an accreditation of a lab from ANAB?
Yeah, so this organization, ANAB, they came into our lab and they reviewed all of our protocols, ranging from facility security to the protocols that we do in the lab to staff education and training, and to make sure that we're meeting all the standards that we are being accredited to.
And the lab at MSPCL, its accreditation, is it up to date at this point?
Yes.
With reference to yourself personally, sir, in regard to your casework, in regard to your training, are you familiar with the term proficiency testing?
Yes.
And can you explain to the jury what that is and sort of how often and what consists of the proficiency testing that you've done?
Yeah, so a proficiency test is given on a semi-annual basis, so twice a year, by an outside agency, and we don't know what the results are, and we treat it as a regular case. So we take it through the regular DNA processing, and we have to pass that proficiency test to show that we maintain our competency in whatever work that we are doing in casework.
And sir, as far as the proficiency testing that you've undergone, what have been the results of your proficiency testing?
I've passed all of mine.
Now, sir, it may be a term that's pretty well understood, but if you could explain to the jury, at least based on your training experience, what your understanding is of a couple of different terms. And first one, I'm going to start with DNA itself. What is DNA?
Yeah, so DNA stands for deoxyribonucleic acid, and that is basically the genetic blueprint that makes us who we are. And we each have 2 copies of it. We inherited one from our mom and one from our dad, and that combination makes us unique. And with the exception of identical twins, everybody has a different DNA.
And is DNA the same in every cell of a person's body?
Yes, barring any rare mutations, DNA is the same. So if you take a blood sample and a saliva sample from the same person, the DNA should be the same.
And what I'm going to ask you now is just a little bit about the process of DNA and how it's tested within your lab. Am I correct in saying that there's essentially 4 steps? Is that correct?
Correct.
And approaching sort of chronologically, one of the steps is called extraction, correct?
Yes.
Can you explain to the jury how it is specifically that DNA is extracted by yourself in general terms when you're testing it?
Yes. So for the extraction step, so I receive the samples. They're in like small tubes. They're closed. I will add a set of reagents to it, apply heat to that sample, and that will actually burst open the cells to make the DNA available for use.
When you are extracting from a sample, do you know, or is there a way to sort of measure how much DNA you're extracting from a sample?
Yes.
And can you explain sort of how it is that you know that?
Yeah, so that's the second step that we call quantitation. And we take a small portion of that DNA that we extracted, and we'll apply a different set of reagents and run it through an instrument that heats and cools down the sample, goes through a cycle, and allows us to estimate how much DNA is extracted.
Now, sir, what is PCR?
PCR is stands for polymerase chain reaction, and it's basically known as molecular xeroxing, where it's making millions of copies of specific areas that you can target of the DNA.
I'm sorry, I didn't mean to step over.
Sorry,
Is that essentially the third step that you undergo?
correct.
Now, what is a STR?
It stands for short tandem repeat, and these are locations in the DNA that change each person. So it's comprised of small little repeats, and each person, however repeats you have at that location, is sort of what will show up in your DNA profile.
Now, how is it with your work in the lab that you identify DNA fragments?
It's the fourth step where we call detection, where after we amplified the specific areas that we are looking at in the DNA, we'll run it through an instrument that will separate them out and generate the DNA profile.
Now, within your work in the lab, are you familiar with terminology as far as a known item versus a question item?
Yes.
And can you explain what each of those mean in reference to your field and your work, and sort of what is the difference between the two?
Yeah, so a known item is an item that's collected directly from an individual and is to be used for comparison purposes, whereas a questioned item is collected from, let's say, like a crime scene where we don't know the source.
Now, as far as your lab or your unit within the lab, are there the DNA unit itself, does it follow sort of standardized policies and protocols?
Yes, we do.
And who approves of those particular policies, protocols, and procedures?
Our technical leader.
Now, could you explain the process used to analyze the evidence in this case?
Yeah, so the evidence in this case underwent that four-step process I just described, and which was extraction to quantitation to amplification, detection, and then interpretation of the profile that was generated.
Now, what are some of the controls, or what do you use when you're during the course of your testing?
Yeah, so there are two types of controls, positive and negative controls. So a positive control is a sample with a known DNA profile that we use to show that each step in the process worked as expected. And a negative control is a sample that has no DNA in it and that only receives the reagents that we are using. And that is to show that there's no inherent contamination of the reagents that we used.
Let me ask you, sir, if you know, what are some of the ways to prevent sort of DNA transfer from yourself or from someone else working on the case or from other samples that you may be working on with others?
Yeah, so there are many ways. One way is to clean the workspace before we start working and after, just to make sure that it's constantly being cleaned. Another way is to wear lab PPE, so personal protective equipment like lab coats, face masks, and hairnets. And another one is to make sure that we only have one tube open at a time so that we don't have any cross-contamination between samples.
Now, your work specifically, sir, does it undergo some sort of review process?
It does.
And specifically your work in this case, did it undergo that same sort of general review process pursuant to the policies and procedures you were speaking about before?
Yes, it did.
Now, when a case comes into the Massachusetts State Police Crime Lab, it's assigned a specific case number, correct?
Correct.
And in this instance, I'm referring to a case number entitled 22-02184. Familiar with that case?
I am.
And you did tested a number of different items within that case, correct?
Correct.
Now, as far as the items are concerned, similar to how the items from a specific investigation or case are assigned a case number, are those different items assigned different item numbers as well?
Yes, they are.
And if there is something that is taken, swabbed, or cut, or something from a specific item, how is that labeled in relation to the item that it was taken from?
So basically, so let's say we have a parent item that's numbered 2-1. And then if we take a cutting of that item, it would be called, if it's the first cutting, it would be called 2-1.1.
Now, in this case, sir, was a known standard process?
Yes, it was.
And can you explain to the jury sort of how that process occurs in regard to processing a known standard?
Yeah, so the lab processing is the same where it undergoes the same 4-step process. And a qualified analyst then looked at the DNA profile that was generated to make sure that it was a single source profile, meaning at each location that we looked at, there were only 1 or 2 peaks. And after that, it underwent the technical and administrative review process before it was closed out and ready for use for comparison.
And do you know whose known DNA standard was processed in this case?
Yes,
And who was it?
a known blood standard from John O'Keefe.
Now, as far as your involvement in this case, what was your involvement in this case?
I performed the testing of the questioned items.
And did you have occasion to perform DNA analysis on certain items submitted to your lab in connection with this case?
Yes, I did.
And let me ask you about some specific items. Were you asked to perform some analysis on a item labeled 3-1.1?
Yes, I was.
And if you know, what was that a sample from?
Would I be able to refer to my notes? Thank you. So that was a sample from passenger side tail light.
And with respect to that, there was a sample that was, or DNA profile that was generated from that swab from the tail light, correct?
Correct.
And what, if anything, were the findings, or what kind of, what was the conclusion of the analysis of the profile from the swab taken from the tail light of the vehicle?
The DNA profile was interpreted as a 3-person mixture, including male DNA.
And to be more specific, when an item comes in, it's assigned an item number. There's also a description that's provided along with sort of where the item came from.
Correct.
And so specifically, item 3-1.1 is a sample from passenger side tail light, and it gives a specific Massachusetts registration plate of 3GC684, correct?
Correct.
And so from the analysis of the swab or the profile that was obtained from the swab from the tail light, what, if anything, was that compared to?
John O'Keefe, standard.
So the sample from Mr. O'Keefe.
Correct.
And with regard to that, is that a comparative analysis that you conducted yourself?
Yes.
And what, if any, opinions or conclusions did you come from as far as the comparative analysis between the swab from the tail light from that vehicle and the profile from Mr. O'Keefe?
The DNA profile from this item is at least 510 non-alien times more likely if it originated from John O'Keefe and 2 unknown individuals than if it originated from 3 unknown, unrelated individuals, and this provides support for an inclusion.
Yeah, now, sir, with reference to that item in that comparative analysis, you said 510 non-alien times, is that correct?
Correct.
And non-alien, just for clarification purposes, do you know how many zeros that is?
It's a 1 followed by 27 zeros.
And with regard to that item coming from, or the profile containing the comparative analysis consistent with the DNA profile of Mr. O'Keefe, there are also 2 unknown contributors, is that correct?
Correct.
Now, with regard to that, are you familiar with that item or the profile from that item being submitted along with some other samples to an outside vendor called Bodhi Technology?
Correct.
Now, with reference to another item contained within that, are you familiar with an item 3-6?
Yes.
And what is item 3-6?
That is the root end of hair from exterior passenger side rear panel.
Now, sir, as far as hair is concerned, describe for the jury a little bit about sort of DNA and how that works between, well, let me ask you this. There's essentially, when it comes to a hair sample, there's typically going to be a root end and a shaft end, correct?
Correct.
And where from the DNA, as your lab is concerned, where is that? Where are you able to sort of test from, or where would you be looking to extract a sample from in order to perform testing?
Yes. So our testing for hairs, the DNA will be coming from any tissue that might be attached to the root end of the hair.
And so you're looking for specifically sort of follicular tissue, is that correct?
Tissues attached to the hair. Yes.
Now, as far as you familiar with the terms sort of autosomal DNA versus mitochondrial DNA?
Yes, I am.
And can you explain for the jury briefly just sort of what your understanding is of each of those two terms and what, if any, differences there are between the two?
Yeah. So autosomal DNA is present in the nucleus of the cell. That is, we inherited that from one copy from our dad, one copy from our mom. As mitochondrial DNA is present within the mitochondria, and that is inherited via the mother.
Now, your lab at the Massachusetts State Police Crime Laboratory, is the lab equipped, or does your lab do mitochondrial DNA testing?
We're not equipped to do mitochondrial testing.
And so when you say that the lab is not equipped to do the testing, what exactly does that mean?
So we don't have the instruments or the facilities to do that testing, and we don't get that testing requested very often. So it makes more sense to send it out to an approved vendor to do that testing.
And one of those approved vendors would be the Voda Technology Lab in Lorton, Virginia, that you were discussing previously.
Correct.
And as far as your understanding is that Voda Technology is equipped and does do mitochondrial DNA testing, correct?
Correct.
Now, with respect to your testing, you did some testing with regard to item 3-6 of the hair, is that correct?
Correct.
And can you explain to the jury sort of what that testing was and what, if any, opinions or conclusions you were able to draw from the testing that you were able to do in the lab?
Yes. So that testing began the same way as the 4-step process. I extracted the hair and then went to quantitation. After quantitation, I determined that there was no human DNA detected. So then testing was halted at that step.
And so what, if anything, was the result of that testing that you conducted with regard to that?
That no human DNA was detected, so STR analysis was not performed.
Now, that phraseology there as far as no human DNA detected, what does that mean?
So quantitation is looking specifically for human DNA, and we couldn't detect that with our instruments. It's not speaking to the hair itself. It's just that we could not detect any human DNA.
That's not so much to say that there is no DNA on the hair, it's just that it is below the level of detection that you're qualified to report. Is that fair to say?
You're correct.
Now, with regard to the taillight sample, if I could step back to that for a second, the 510 nonillion times, what, if any, relationship does that have to sort of the overall world human population?
So the world population, I think, currently is about 8 billion, which is an 8 followed by 9 zeros. And 510 nonillion has 30 zeros in it. So it's larger.
Significantly more. Correct?
Significantly larger, sir. Yes.
Now, sir, if I could turn your attention to some other items that you tested in relation to this case. And just to be clear, sir, you conducted or issued about 6 different DNA reports in regard to the testing that you conducted in this case. Is that correct?
Correct.
So if I could direct your attention now to the 6 reported DNA testing report number 6. That involved an item called 3-3.1. Is that correct?
Correct.
And what is the description of that item?
Oh, wait, I'm sorry. Got the wrong report. That is a sample from apparent glass on rear bumper.
And with the same mass registration plate as the taillight, is that correct?
Correct.
And what, if any, conclusions or opinions were you able to draw from your analysis of the testing on that particular item?
So at the quantitation step for this item, insufficient human DNA was detected and STR analysis was not performed.
And again, just as far as that statement is concerned, can you explain a little bit to the jury what that means as far as what your opinions or what you're seeing in regard to insufficient human DNA detected and STR analysis not performed?
Yes, so we do at the quantitation step, we do have a cutoff, a certain value where if we detect human DNA that's below this cutoff, we validated that it's not going to, most likely not going to provide a profile that can be used. So we halt testing at that step.
Now, sir, if I could direct your attention to the fifth report that you issued in this case, DNA testing report number 5. And does that sort of contain the totality of the items that you analyzed in this particular case?
It does.
Now, sir, if I could direct your attention to an item labeled as number 3-2.1.1. And what is the description for that particular item, sir?
That is a sample from exterior of broken drinking glass.
And with reference, were you able to generate a DNA profile from the swab with the sampling from that item?
Yes.
And what, if any, comparison were you able to do in regard to the DNA profile of Mr. O'Keefe?
Yes, so the sample was interpreted as a mixture of 3 contributors, and the DNA profile for this item is at least 530 non-alien times more likely if it originated from John O'Keefe and 2 unknown individuals than if it originated from 3 unknown unrelated individuals, and this provides support for an inclusion.
And so the sample from the exterior of the broken drinking glass is at least 530 non-alien times more likely if it originated from John O'Keefe, correct?
Correct.
Now, if I could turn your attention to item 7-1.2.1. And what is the description associated with that item?
That is a sample from stain A on upper right leg of jeans.
And were you able to generate a DNA profile from the sample from what's labeled as stain A on the upper right leg of Mr. O'Keefe's jeans?
I was.
And what, if any, comparison were you able to do with relation to that profile from that sample to Mr. O'Keefe's DNA profile?
Yes, so the DNA profile was interpreted as a mixture of 3 contributors, including male DNA. Contributor 1 was suitable for comparison, and due to limited information, contributors 2 and 3 were not suitable for comparisons. And the DNA profile from this item is at least 570 non-alien times more likely if it originated from John O'Keefe and 2 unknown individuals than if it originated from 3 unknown individuals. And this provides support for an inclusion.
And so when you say, as far as due to limited information, contributors 2 and 3 are not suitable for comparison, what does that mean?
So that just means that those contributors were very low and that any comparisons that would be done to them could not be reliable based on our protocols and validations.
And so from your comparative analysis, you came to the opinion or conclusion that the sample from the upper right leg of Mr. O'Keefe's jeans was 570 non-alien times more likely than it originated from Mr. O'Keefe, correct?
Could you rephrase that?
With respect to your comparative analysis of the profile from the sample from stain A on the upper right leg of Mr. O'Keefe's jeans, your opinion is that it was at least 570 non-alien times more likely that it originated from John O'Keefe, correct?
From then,
System.
What was your conclusion as it came to item 7-1.2.1?
so the DNA profile from this item is at least 570 non-alien times more likely if it originated from John O'Keefe and 2 unknown individuals than if it originated from 3 unknown, unrelated individuals, and this provides support for an inclusion.
Sir, let me break from this for a second, and I just want to ask you a little bit about transfer DNA. Are you familiar with that term?
Yes.
And can you explain to the jury what you understand that term means?
So transfer DNA just means DNA that sort of transfers based on just some activity that can happen. Like if somebody touches a water bottle, they might transfer some DNA onto that water bottle.
Now, from the fact that DNA is on something or on an item and it matches a profile from an individual, is there anything from that that you can opine or conclude as to how that DNA was deposited or when?
No, I cannot.
Now, with reference to factors that may cause a degradation of a DNA sample or a touch DNA sample that's left, what, if any, are some of the factors that may affect that as far as causing degradation of a sample or you're causing an inability for you to be able to generate a DNA profile from another?
Some factors include being exposed to heat for a prolonged time and ultraviolet rays from the sun, as well as just time. It might eventually lead to some degradation.
Now, as far as items that are outside, Because you indicated that being hot or warm temperatures might have some effect on it. What, if any, impact will cold temperatures have?
So we store our DNA extracts in a cold room. So cold temperatures could be very good to help preserve any DNA that's on there.
Now, if I could turn your attention back to your report number 5 and ask you with reference to item 7-1.3.1. And can you, what is the description for that item, sir?
That is a sample from stain B on upper right leg of genes.
And again, those would be the genes of Mr. O'Keefe, is that right?
Correct.
And you were able to generate a DNA profile from that sample as well?
Correct.
And what, if any, comparison were you able to do between the profile from that DNA on the upper right leg of Mr. O'Keefe's genes and Mr. O'Keefe's DNA?
Yeah, so the DNA profile was interpreted as a mixture of 3 contributors, including male DNA. And the DNA profile from this item is at least 660 nonalien times more likely if it originated from John O'Keefe and 2 unknown individuals than if it originated from 3 unknown, unrelated individuals. And this provides support for an inclusion.
Next, if I could turn your attention to item 7-1.4.1, and what is that sample, or what is that item or sample from?
That is a sample from stain C on upper right leg of genes.
And those genes would be Mr. O'Keefe's genes as well?
Correct.
And were you able to then generate a DNA profile from that sample?
Yes.
And what, if any, comparison or what, if any, conclusions or opinions did you draw from a comparative analysis of the DNA profile from that sample from the upper right leg of Mr. O'Keefe's genes and Mr. O'Keefe's DNA profile?
So the DNA profile is a mixture of at least 2 contributors, including male DNA. Contributor 1 is suitable for comparison, and due to limited information, contributor 2 is not suitable for comparison. The DNA profile was interpreted as a mixture of 2 contributors, and the DNA profile of this item is at least 830 nonalien times more likely if it originated from John O'Keefe and an unknown individual than if it originated from 2 unknown, unrelated individuals, and this provides support for an inclusion.
Now, sir, if I could turn your attention to item 7-1.5.1, and what was where did that come from?
That is sample from stain D on upper right leg of genes.
And again, were you able to generate a DNA profile for that sample?
Yes.
And what, if anything, were your findings in relation to comparative analysis between that and that DNA profile from that sample and the DNA profile of Mr. O'Keefe?
The DNA profile was interpreted as a mixture of 3 contributors, including male DNA. And the DNA profile from this item is at least 670 nonalien times more likely if it originated from John O'Keefe and 2 unknown individuals than if it originated from 3 unknown, unrelated individuals. And this provides support for an inclusion.
Now, sir, if I could turn your attention to item 7-1.6.1. And what is the description for that item?
That is sample from stain E on upper right leg of genes.
And were you able to also generate a DNA profile from that sample?
Yes.
And did you then conduct a comparative analysis between the DNA profile from that sample and the DNA profile from Mr. O'Keefe?
Yes.
And what, if any, conclusions or opinions or findings did you have in regard to that comparative analysis?
Yeah, so the DNA profile was interpreted as a mixture of 2 contributors, including male DNA. Contributor 1 is suitable for comparison, and due to limited information, contributor 2 is not suitable for comparison. The DNA profile from this item is at least 1 decillion times more likely if it originated from John O'Keefe and an unknown individual than if it originated from 2 unknown, unrelated individuals, and this provides support for an inclusion.
Now, as far as that decillion, again, similar to the nonalien, how many zeros are we talking about?
That's a 1 followed by 33 zeros.
Now, if I could turn your attention to item 7-1.7.1. You familiar with that item, sir?
Yes.
And what is the description for that item?
That is sample from unstained areas of exterior of genes.
And were you able to generate a DNA profile from that sample?
Yes.
And similarly, were you able to then do a comparative analysis between the DNA profile from that sample and the DNA profile of Mr. O'Keefe?
Yes.
And what, if anything, were your findings in regard to that comparative analysis?
Yep. The DNA profile was interpreted as a mixture of 4 contributors, including male DNA. The DNA profile from this item is at least 553 septillion times more likely if it originated from John O'Keefe and 3 unknown individuals than if it originated from 4 unknown, unrelated individuals. This provides support for an inclusion.
And ultimately, sir, with regard to that number, septillion, how many zeros is that?
That is a 1 followed by 24 zeros.
I'm going to ask some questions about a number of different items from 7-17. These are samples taken from an orange T-shirt as well as a gray long sleeve shirt. Is that correct?
Correct.
And if you could, essentially you did testing on those items as well as some fingernail clippings belonging to Mr. O'Keefe,
Correct.
or samples from the fingernail clippings.
Yes.
Now, with regard to those items, swabbings taken from the stains from various areas of the orange T-shirt, and just in general, where were those areas on the T-shirt, the orange T-shirt to start with, that the swabbings were taken from?
The top front left of orange shirt and front bottom right of the shirt as well, and just the back left shoulder of the orange shirt.
Now, with regard to the gray long sleeve shirt, where are the areas on the shirt where those swabbings were taken?
The right sleeve, the center front, the back left, the back right, and the back bottom of right.
And were you able to generate DNA profiles from each of those items?
I was.
And were you then able to do a comparative analysis between the DNA profile from each of those items with relation to the DNA profile from Mr. O'Keefe?
I was.
And what, if any, conclusions were you able to draw from that comparative analysis?
So the male DNA profile was interpreted as originating from a single contributor, and the DNA profile from these items is at least 490 octillion times more likely if it originated from John O'Keefe than if it originated from an unknown, unrelated individual. This provides support for an inclusion.
As far as that number is concerned, octillion, how many zeros is in an octillion?
That's a 1 followed by 27 zeros.
And with regard to each of those items, from the orange T-shirt, the gray long sleeve T-shirt, and the fingernail clippings of, they were taken from both the right and left hands of Mr. O'Keefe, is that correct?
Correct.
So that's 490 followed by 27 zeros. That would be from a single contributor, correct?
Correct.
Now, sir, if I could turn your attention to item 7-18, I'm sorry, 7-18.4.1. You're familiar with that item?
Yes.
And what is the description associated with that item?
Sample from stain C on top front gray long sleeve shirt.
And were you able to generate a DNA profile with respect to that item?
Yes.
And were you able to then do a comparative analysis between that DNA profile from that item and the DNA profile of Mr. O'Keefe?
Yes, I was.
And what, if any, conclusions did you come to with regard to that?
The male DNA profile was interpreted as originating from a single contributor. The DNA profile from this item is at least 58 octillion times more likely if it originated from John O'Keefe than if it originated from an unknown, unrelated individual. This provides support for an inclusion.
And with regard to that stain from the gray long sleeve T-shirt, again, that was determined by you through your testing to be DNA from a single contributor matched John O'Keefe, correct?
It was determined to be from a single contributor, yes.
Now, sir, if I could turn your attention to item 7-18.14.1. Are you familiar with that item?
Yes.
And what is the description associated with that item, sir?
A sample from stain L on back bottom right of gray long sleeve shirt.
And were you able to generate a DNA profile from that, sample from that item as well?
Yes, I was.
And were you able to then do a comparative analysis between the sample, the profile from that sample versus the profile from Mr. O'Keefe?
Yes, I was.
And what, if anything, were your findings in relation to?
So the DNA profile was interpreted as a mixture of two contributors, including male DNA. The DNA profile from this item is at least 1 decillion times more likely if it originated from John O'Keefe and an unknown individual than if it originated from two unknown, unrelated individuals. This provides support for an inclusion.
And sir, I'm going to ask you, or draw your attention to item 7-18.17.1. Familiar with that item?
Yes.
And what is the description associated with the sample from that item?
Sample from unstained areas of exterior of gray long sleeve shirt.
And were you able to also generate a DNA profile from that sample?
Yes.
And what, if any, were you able to then do a comparative analysis between the sample profile from that sample and the profile from Mr. O'Keefe?
I was.
And what, if any, conclusions did you come to with reference to that comparative analysis?
The DNA profile was interpreted as a mixture of 2 contributors, including male DNA. Contributor 1 is suitable for comparison, and due to limited information, contributor 2 is not suitable for comparison. The DNA profile from this item is at least 960 non-million times more likely if it originated from John O'Keefe and an unknown individual than if it originated from 2 unknown, unrelated individuals. This provides support for an inclusion.
Sir, there are some other items from which you conducted testing but were not able to generate a profile from, correct?
Correct.
And specifically, I'm going to ask about item 7-3.1. That's a sample from the right instep of Mr. O'Keefe's sneaker, is that correct?
Could you repeat that number again, please?
Sir, if I could direct your attention within your DNA report number 5, I'm talking about item number 9.
Yep.
And so, sir, with reference to that, what, if any, is an indication as far as the mixture not being suitable for comparison due to the quality of the profile, correct?
Correct. It was a mixture of at least 5 contributors.
And so from that, as far as not suitable for comparison, can you explain to the jury what that means?
Yes. So during our validations, we've only validated mixtures up to 4 contributors because we've determined that once you get more than 4, like at least 5 contributors into a mixture, it becomes too complex to be able to make any reliable comparisons to that mixture.
Sir, if I could direct you to item number 11 or item number 7-18.3.1 within your report.
Right. Yep.
And that's a stain from the gray long sleeve t-shirt top front, is that correct?
Yes.
Now that is indicated as not suitable for comparison due to the quality of the profile, correct?
Correct.
And can you explain to the jury what that means as far as not being suitable due to the quality of the profile?
Yes. So during our bringing it back to our 3rd step of amplification, where we're making millions of copies of specific locations, sometimes there might not be a lot of DNA there, and that amplification step might not amplify all the information that could be present, and the profile that's generated won't have all the information to be able to reliably make a comparison. So the information that we are able to look at is very limited, so we just do not do any comparisons because they would not be reliable.
And was that same as far as not suitable for comparison due to the quality of the profile? Was that the same for 2 other areas that you tested in regard to the front or sample from the front of the gray long sleeve shirt and a sample from the back left sleeve of the gray long sleeve shirt?
Yes.
May I have a moment?
Yes.
Sir, let me ask you just briefly, you're familiar with the term called SDR mix?
Yes.
And can you explain to the jury what that is and how that is involved or how it's implicated within your test?
Yeah. So StarMix is a tool that we use to basically break down complex mixtures into its individual contributors and into like a way that it would best describe that mixture. And once it does that, it'll then do comparisons and calculate a likelihood ratio, which is a statistical weight to that comparison.
And how does that relate to the testing that you did in this case?
I used StarMix on all the samples that had comparisons done to them.
Thank you, sir. Thank you, sir. I have no further questions, Your Honor.
Ms. Little? No questions. Thank you. All right, Mr. Porto, you are all set, sir.
Thank you.
Could I see counsel regarding scheduling, please?
Yes, Your Honor.
please.
So we're actually going to send you home today because we're on schedule. It's beautiful out. Rather than start with the witness that we'll start with tomorrow morning, it makes sense. I'm going to talk to the lawyers for a few minutes. So we'll send you home. Do not discuss this case with anyone. And just so that I'm clear, that means each other. That means any aspect of this case, any observations, anything at all. Do not do any independent research or investigation into this case. If you happen to see, hear, or read anything about this case, please disregard it. We expect a full day tomorrow. So we'll see you tomorrow.
All rise for the court, please.
Sir, follow me, please.
So I'm just going to ask, I'm going to talk about that now.
Thank you. Please be seated. Court remains in session.
All right, so I'll see counsel at sidebar about a couple of issues.